Hello students, welcome to lecture series on agrochemicals and pest control. In this episode, we will be studying about plant pathology. The word pathology has been derived from two Greek words wherein pathos means suffering and logy means discuss or to speak. Plant pathology, also called as phytopathology, represents the area of biological sciences which concerns with the suffering of plants. Plant diseases are caused by biotic agents like fungi, bacteria, actinomycetes, mycoplasma, viruses, nematodes and flowering parasites, or by abiotic factors like unfavorable environmental conditions, nutritional deficiencies. Study of plant pathology includes the study of sciences via microbiology, bacteriology, virology, mycology, nematology, protozoanology, phycology, unfavorable environmental factors, nutritional deficiencies and flowering plant parasites. Plant pathology is one among the branches of agricultural sciences that deals with the causes, etiology, resulting losses and management of plant diseases with four major objectives. They are study of the diseases or disorders caused by biotic and abiotic agents, study of mechanism of disease development by pathogens, study of interaction between plant and pathogen in relation to the overall environment, and develop suitable management strategies for managing the diseases and losses caused by them. Hence, in this episode, the following aspects are going to be seen. They are history of plant pathology, the disease triangle, disease diagnosis, disease development and plant disease control. We shall move to the first aspect, history of plant pathology. By studying the history of science, we get a better perspective of the subject. We come to know the contributions made in that field, the problems that are encountered and the manner in which they are tackled. The history of plant pathology is divided into 5 different eras. They are ancient era, ancient to 5th century, that is 476 AD. Secondly, dark era, 5th to 16th century, that is 476 AD to 1600. Next is pre-modern era, 17th century to 1853, that is 1600 to 1853. Next is modern era, 1853 to 1906 and present era, that is 1906 onwards. Moving to the first era, that is ancient era, ancient to 5th century, that is 476 AD. Diseases in plant have been known since ancient times. Rust, blight, mildews, smuts were familiar to Hebrew, Greeks, Romans, Chinese and Indians. Plant diseases was recorded in Rig Veda, Atharva Veda as early as 1200 BC. Symptoms and control of diseases have been mentioned in Riksha Ayurveda by Surpal in ancient India. Even mention of plant diseases have been made in Buddhist literature of 500 BC. Theophrastus in 300 BC, a great botanist, noted occurrence of crop diseases and suggested some remedies to control them. He also wrote about plant diseases in that era. In 100 AD, Lord Pliny described plant diseases and suggested some remedies. He believed that disease originated from plants or from the environment. Next era is dark era, that is 476 AD to 1600. Plant pathology made very little progress during this era. Some Arabians like Ibn al-Awan described symptoms and control measures for some plant diseases. Next is pre-modern era, 17th century to 1853, that is 1600 to 1853. There are several scientists who contributed to plant pathology. They are Robert Hooke from 1665. He is called the father of cell theory and he invented first compound microscope. He reported that plant tissues are made up of minute units called cells. Next is Anton van Leeuwenhoek, 1676. A Dutch worker from Holland, he invented first simple microscope with home ground lenses between two metal plates. He described different types of protozoan and bacteria as little animalcules. All unicellular microorganisms were first recorded by him. Next is P. A. Micheli, 1729. An Italian botanist studied several fungi and described their morphology for the first time. He proposed that fungi originated from spores and he was regarded as founder of mycology. Next is John Needham, 1743. He reported plant parasitic nematodes in wheat galls. Next is Carolus Linnaeus, 1753. He established Latin binomial system of nomenclature of plants and animals in his book Species Plantarium. Next is Prevost, 1807. He is a French botanist suggested copper sulfate seed treatment for bunt of wheat. Coming to the next era, that is modern era, 1853 to 1906. This known as pathogenic period, which was devoted to the study of role of fungi causing plant diseases. Here comes some scientists like Anton D. Barry, 1853. He proved that late blight of potato was caused by Phytophthora infestans. He is known as father of plant pathology. Next is T. J. Burkill, 1873. American plant pathologist, and he proved bacterial nature of fire blight of apple and pear. Next is Robert Koch, 1876. He found bacterial nature of anthrax disease in animals in 1881. Gelatin was used as solidifying agent in culture media and it was replaced by agar-agar. He described the theory called Koch postulates. Next is P. A. Millardet, 1882 to 1885. He used Bordeaux's mixture, that is copper sulfate plus lime, for control of downy mildew of grapes. Next is Adolf Meyer, 1886. He described tobacco mosaic virus, which is abbreviated as TMV, and proved that tobacco mosaic virus shall be transmitted from diseased plant to healthy plants. Next is E. F. Smith, 1870. Father of phytobacteriology, he worked on bacterial wilt of cucurbits and crown gall diseases. Next is Ivanovsky, 1892. He demonstrated that tobacco mosaic virus can pass through bacteria-proof filters and proved filterable nature of viruses. Next is Craigie, 1897. He showed function of Puccinia in rust fungi. Next is Biffen, 1905. He is the pioneer in genetics of plant disease resistance. The next era is present era, that is from 1906 onwards. The present or the current era commencing from 1906 has since remarkable discoveries. Some of the scientists under this era are J. C. Luthra, that is from 1931. He used solar heat treatment for loose mud of wheat. Next is W. M. Stanley, 1935. He proved crystalline nature of viruses and he also got Nobel Prize. F.C. Bowden and Perry in 1936. They proved nucleoproteinaceous nature of viruses. G.H. Flohr 1955. He explained the gene for gene theory hypothesis. Last is Doi and Asuyama. They discovered mycoplasma like organism, which is also called as MLO, responsible for Yell's type of disease. We shall move to the second aspect, the disease triangle. Plant disease results when a specific agent such as persistent unfavorable environmental conditions or the activity of a pathogen disrupts physiological functions, causing plants to deviate from normal development. The word persistent is used to distinguish between a disease which develops over time and an injury which occurs more or less instantaneously. Non-infectious diseases, perhaps more appropriately called disorders, do not spread from plant to plant. These disorders results from the plant's exposure to such factors as unfavorable weather, mechanical damage, nutrient deficiencies, excess salts or toxic chemicals. Although disorders can predispose plants to infection by pathogens, disorders are not directly treated using pesticides. Here we emphasize on the infectious plant diseases caused by pathogens. An infectious disease results when a pathogen lives in close association with the host plant. The pathogen is functioning as a parasite, since this relationship usually benefits the pathogen at the host expense. Although relationships resulting in disease are often complex, three critical factors must be present in order for a particular disease to result. The three factors necessary for disease development are: the pathogen must be present, a susceptible host must be present, and the proper environment must be present permitting infection of the host. These factors give us the concept of the disease triangle. Production practices followed by large growers and home gardeners serve to modify the interaction of these three factors to reduce or unwittingly increase the overall impact and severity of plant diseases. Plant professionals use the disease triangle concept to simplify remembering and understanding how a particular disease develops and why various control strategies are used. Next is the pathogen. The presence or the absence of a pathogen is the main factor that determines whether disease occurs. Introduction of a pathogen to an area from which it has previously been absent can cause major outbreaks of disease in plant communities. The amount of disease that develops is often determined by the pathogenicity of the main pathogen. The term pathogenicity relates to both the virulence and the aggressiveness of the pathogen. Pathogenicity is dependent on the pathogen's reproductive, dispersal and survival fitness. The adaptability of the pathogen is also important in determining its ability to infect resistant host or to survive changed environmental conditions. Adaptability is determined by the pathogen's genetic flexibility and reproductive efficiency. The spread of a disease and the formation of epidemics are reliant on the pathogen's ability to disperse rapidly over long distances. Next is the host. Levels of resistance or susceptibility of the host plant will affect disease development. Plants with high level of resistance are essentially immune or not susceptible and do not allow the pathogen to become established even if present. Plants with lower levels of resistance may become infected but only allow the pathogen to develop slowly, limiting the economic impact of the disease to acceptable levels. Stress such that brought on by an unfavorable environment, poor fertility or irrigation will affect a host plant's ability to resist infection and therefore also affects disease development. In some cases, resistance in the host is overcome by the pathogen, resulting in rapid and devastating disease development. This risk is increased if large production areas are planted to a monoculture of identical host. Therefore, genetic diversity of host plants will decrease the risk of catastrophic levels. Next is the environment. The presence of a pathogen against a particular plant will generally not cause serious disease unless the environmental conditions are favorable. This includes the aerial environment and the soil environment. Human attempts at controlling disease usually involve manipulating the environment in some way. For example, breeding wheat cultivars to tolerant dry conditions allow Australian farmers to plant the crop in areas that are not favorable for pathogens such as powdery mildew and leaf rust. Properties of the aerial environment that influence disease development include moisture levels, temperature and pollution. Moisture is particularly important to pathogenic bacteria and fungi. Rain splash plays an important role in the dispersal of some fungi and nearly all bacteria, and a period of leaf wetness is necessary for the germination of most airborne spores. Temperature also affects the incubation or the latent period, the generation time and the infectious period. The disease cycle speeds up at higher temperatures, resulting in faster development or epidemics. A high concentration of pollutants can affect disease development and in extreme cases damage the plants directly by causing acid rain. The soil environment affects soil-borne diseases largely by determining the amount of moisture available to pathogens for germination, survival and motility. Germination and infection success also rely on the temperature of the soil. The fertility and organic matter content of the soil can affect the development of the disease. We shall move into the third aspect, disease diagnosis. Diagnosis requires knowledge of what is normal for the host plant as well as knowledge of problems that occurred in the past. Accurate diagnosis is critical for deciding if a disease is present and if effective control measures are available or justified. An accurate diagnosis is based upon recognition of specific signs of the pathogen on the host as well as the presence of symptoms on the host. Signs are defined as a visible presence of the pathogen or products of the pathogens on the host plant. Fungal spores, fruiting structures that bear spores and bacterial oozes are all examples of signs. In contrast, symptoms are the external and internal reactions or alterations of a plant as a result of disease. For example, dead spots in leaves or bark, rotten spots in fruits or tubers, swellings on roots or branches, clustered bunches, unnatural color or shape and vascular discoloration in stems are all the symptoms that may aid in diagnosis of a plant diseases. We shall move into the fourth aspect, disease development. It is important to understand how plant diseases develop in order to control them. By the time it becomes obvious that a plant has a disease, it is generally too late to do anything about it in that growing season. Plants cannot be cured in the way people expect their own illness to be cured. The process by which disease develops can be broken into 5 distinct phases. They are inoculation. This is the introduction of the pathogen to the host plant tissue. Wind or rain or running water can move pathogens and introduce them into the host plant. Some pathogens move themselves short distance, but most rely on the other means such as birds, insects, people or equipments. Sources of inoculum include plant debris, seed, perennial plants and soil. Next is incubation. This is a period of development during which the pathogen undergoes changes to develop a form which can penetrate or infect the new host plant. Some fungi, for instance, grow a structure called a penetration peg that can grow through the cell walls of the plant. Next is penetration. This is the process of getting inside the plant. It may be an active or passive process. Some pathogens produce enzymes to dissolve the cutin and cellulose layers of the plant material between them and the cell content. Some pathogens can swim through water on the plant surface and into the plant through natural openings such as stomata, lenticels or hydatodes or through wounds. Some pathogens are put inside the plant by insects, pruning tools or driving rain. Next is infection. When the pathogens invades the plant tissues and establish a parasitic relationship between itself and the host, infection has occurred. Next is disease. When the host plant responds to the presence of a pathogen, a disease exists. The host response results in symptoms of the disease such as blight or necrosis. As the pathogen matures, it produces inoculum in the form of spores, virus particles and bacterial cells that can spread or disseminated to other adjacent healthy plants. We shall move into the last aspect, plant disease control. The importance of understanding the disease development process becomes obvious when considering control options. By the time symptoms are expressed, the pathogen, with few exceptions, is already inside the host plant and is relatively safe. Therefore, control efforts in most cases must occur before penetration has taken place. The overall principle in effective disease control is to keep the inoculum density of the pathogen at a very low levels. Success in controlling plant disease will occur when combinations of the following methods of control are used. They are avoidance. A grower can avoid certain diseases by choice of geographical area or choice of planting site. Disease can be avoided by planting at a time that does not favor disease. Using disease-free planting stocks or modifying cultural practices also helps to avoid disease. Next is exclusion. A grower can inspect stock of signs of diseases and reject or treat any which is suspect. Plant quarantines are designed to exclude certain pests from areas that are free of that pest. Elimination of carrier insects can exclude a disease. Next is eradication. Once a disease is established in an area, eradication is unlikely. However, significant reduction in disease inoculum can be attained by destroying diseased plant or alternative host by rotating crops or by soil solarization. Next is protection. Spraying or dusting plants with fungicides or bactericides can protect them from disease. Sometimes modifying cultural practices or the environment may protect the crop. Control of carrier insects will also protect the plants. Next is resistance. Breeding and selection are used to develop resistant crops. Resistance can be enhanced through proper culture of a crop. Tolerance is another form of resistance in which the plant becomes infected but goes on to mature and yield normally. Next is sanitation. Several disinfectants can be used to surface sterilize growing surfaces. Bleach in 1:1 solution is effective against viruses, bacteria and fungal spores. It is not effective against sclerotia or other resting fungal structures. Alcohol may be used as a disinfectant of tools, but it is not effective against fungal sclerotia, viruses and some bacteria. Next is therapy. Removal of diseased parts of a plant will sometimes control the disease. Heat can be used to treat contaminated seeds and to eliminate viruses from certain type of fruit tree bud wood. Familiarity with crops and the diseases and insects that affect them is useful in planning control programs. Some diseases occur every season, others occur sporadically. Some can be controlled easily by using proper methods, others must be tolerated. Knowing which problem falls into which category comes with experience. Knowing the proper method to use at the proper time is a part of integrated pest management. We shall conclude, plants are constantly confronted with a wide variety of potential pathogens within their environment. Nevertheless, the development of disease is the exception rather than the rule, due to the high efficient nature of the coordinated system of passive and active defenses that have evolved in the plants. These defenses generally limit the host range of a microorganism capable of causing plant diseases to the members of a single plant genus or species and affect all races of a particular pathogen similarly. However, plant disease resistance can also be induced in specific plant cultivars within the host range for only in. Thank you.